My name is Bas Trietsch. I'm the Chief Technology Officer at MIMETAS. I'm one of the co-founders, so I've been with the company now for about 7 years. MIMETAS is a biotech company where we are developing new therapies by using organ-on-a-chip technology. So that means that we are growing miniature versions of organs such as liver, kidney, lung, inside microfluidic chips that we use to be able to more quickly design new therapies for patients. So the therapies we are developing span a wide range of diseases, and typically we are focusing on the more complex diseases because that's where having the best model systems that really recapitulate how the body responds to potential new drugs are needed to understand how a disease works and also to find the best new therapies to really help impact the lives of patients. The basic technology that we use is the organoplate. This is a platform technology which is basically a plastic plate about that size, and in one of those plates we can culture up to 100 miniature livers or 100 miniature kidneys or even combinations of different organs. So that means that on a given day we typically have a couple of tens of thousands of miniature organs that all of our researchers can perform their tests on, and you can imagine that that results in much quicker development of new therapies, and all of that we can do without needing also tens of thousands of laboratory animals. We take a system in which we can really mimic organs and tissues as they are in the body without sacrificing that throughput. And I think that combination of high throughput and high fidelity biology, really best in class biology, gives us the edge and gives us a unique opportunity to solve diseases, to find treatments that are otherwise unattainable. Yeah, so I think BioPartner 5 is a very nice place to be in, and I think there's a nice correlation between the fact that one of the earliest incentives for MIMETAS to start to build this organ-on-a-chip technology was not only to have better models to gain better understanding of how diseases work, but also to be able to do that without using too many laboratory animals. And I think that that ethical drive is something that is very much represented in this building as well. And it's actually quite fun that the place where I did my studies, where I was doing my PhD when we founded the company, is actually the Gorleus laboratory, which is now partially replaced, and a lot of the material that came out of that building has followed us all the way into this new and shiny new building. I think a lot of the steel beams here are actually the same beams as the one where I manually made the first organoplate and pipetted the first experiment. So I think it has a nice circularity to it where we stay within this very inspiring environment and have a sense of history attached as well.